All right. I hope you're all ready to think a bit about diversity in morphology of phytoplankton. As you can see from my title, I'm going to talk about some results that rely on a very large data set. And right out of the gate, I want to acknowledge the extreme hard work and dedication of my co-authors, and actually a small army of other people who have contributed in large and small ways to making sure that we could collect this data set, which has been more than 15 years in the making. So I want to start by bringing right to the front of your minds that many phytoplankton are not little spheroids. They're not even they're not little spheres, and they're not even little spheroids. We know that they vary incredibly in size, many orders of magnitude, but they also vary greatly in shape. And this diversity in shape and size, as we know, has huge impacts on their function, everything from absorption of light to acquisition of nutrients to interactions with grazers, encounter rates with parasites, interactions with flow fields, et cetera. All of these incredibly important matters of life and death in the plankton depend on variations in size and shape. These images that I'm showing you on this slide to convey this point come from imaging FlowCytobot, this technology you see here, the workhorse of the data set I'm going to talk about. So I will explain a bit more about it in a few minutes. So, of course, the importance of morphological variability in the phytoplankton is not a new idea. It's been considered for a long time. And I thought I would have a little bit of fun. Oh, it's fun for me. I hope it's a teeny bit fun for you too. With this talk, stepping through a long period, 3 what I think are thought-provoking papers on this topic that are each about 20 years apart, and kind of revisiting the ideas and the approaches in these papers in the context of this new data set. So I want to start with this seminal paper by Lewis in 1976. Lewis proposed this 2-axis trait framework, a combination of important factors. Just 1 minute, let me get rid of this. And variability and trade-offs that is, I think, very compact way of looking at important aspects of the size shape envelopes that phytoplankton occupy. So these kind of, I'll call them iso-shaped lines in this schematic, show the trade-off between maximum linear dimension on the y-axis and surface area to volume on the x for various shapes, where the leftmost diagonal line labeled 1 shows the dependence for spherical shapes, which is, as we know, kind of the extreme worst case scenario for a given maximum length in terms of optimizing surface to volume. What Lewis did is he analyzed 25 species of phytoplankton and he placed them into this framework. And he postulated that evolutionary or adaptive processes have shaped this envelope of realized shapes that he found for these phytoplankton, the dominant taxa in, I think it was a tropical lake. A major point I want you to take away from this is there's huge deviations from sphericity, right? We love these shorthands like equivalent spherical diameter or just using the biovolume of cells as indicator of their size differences. But these metrics are really limited in important ways. And I'm always reminded of the physicist joke, imagine a spherical cow, which usually makes us kind of laugh, like that's a totally maladaptive animal, but we don't think twice about imagining a spherical phytoplankton. It's equally as ridiculous, I would argue. Okay, so Lewis concluded that the phytoplankton surface area to volume ratios are conserved within a much narrower range than you would expect if you just made a random choice of shapes. And he suggested that these constraints can give us insights into the adaptive regulation. He also, of course, pointed out that we needed more studies to confirm these constrained ranges. So this brings me to, okay, what do we find if we look at a few more than 25 points? Was Lewis onto something? So for this, we can turn to the image data that we've been collecting from IFCB in this case deployed at the Martha's Vineyard Coastal Observatory. This is a cabled site off the coast of the Northeast US, where we've been deploying IFCB since 2006. No 20 there. It automatically samples the ocean. It's basically an underwater microscope and makes thousands of measurements of individual cells every hour. And we started this in 2006 and have been continuing it to today. In fact, data is being collected while I'm speaking to you. And as I said, it collects samples every multiple times an hour, thousands of images each sample. So at this point, this data set is more than 1 billion images and approximately 280,000 times that we've sampled the ocean. Okay, so what do we do with all of these images to be able to, I wanted to just take a step aside for a minute to talk about the way the images are measured to convey how useful I think they are for characterizing morphology. This instrument is a flow cytometer in the conventional sense that the sample's injected into the measurement flow cell in the center of a carrier, a particle-free carrier fluid, what we call the sheath, so that it's drawn out into a long, thin stream, and the particles, the cells are going single file through the measurement area. They go first through a laser beam, where they generate a trigger signal, and then a fraction of a, at that point we know there's a cell there, so a fraction of a second later, we flash a strobe, grab a frame from a video camera, and we get a picture that's been triggered. So we only take a picture when we know there's a target there. And because of the controlled flow, this is a replay of an actual sample that's flowing down. The laser beam's like above the screen. And the black box is the camera field. In real time, we're finding the region of the camera field that contains the target and only saving that. So that's what you're seeing here. And in the real sample, they're coming along one at a time, but I've held them for a second in the replay, a few seconds in the replay, so you can see them before they go by too fast. So the point is sheath on the edge, particles in the center, they're aligned with the flow, they're in the focal plane, and they sort of lay flat in the flow so we get the largest cross-section imaged. So I'm not cherry picking when I show these images. The quality is pretty good all the time. Okay, so what do we do with all these thousands of images an hour? Of course, we need automated processing. We've developed algorithms to get geometric characters from all of these complex images. Here I'm just showing the maximum linear extent, in this case determined by ferrite diameter. For the Lewis framework, of course, we want some information about the three-dimensional structure of the cells. For something like that long diatom chain at the top, it's pretty easy to get the major axis into a solid revolution and estimate 3D volume, no problem. But complex shapes like the diatom colony and this dinoflagellate I'm showing here on the bottom, you can't do a simple calculation like that. So fortunately, I worked with a very creative student about 10 years ago who developed this clever approach using a distance map calculation. Distance map is a very simple concept. It's just for every pixel in the target, it's the distance to the nearest boundary of the perimeter image. So you can see those color-coded here for these different shapes. And then as she showed in her paper, it's a pretty simple arithmetic to calculate an estimate of the three-dimensional shape that can be inferred from some assumptions about these distance maps. I'm going to show you a bit about what this looks like for these two complicated examples. We're basically doing like a digital blow-up of these of the shapes, and there's assumptions here. It's not perfect, but this is like light years better than looking through a microscope, writing down two numbers for 10 species, and then estimating volume from those. Okay, so we don't really make these 3D renderings for every cell. I'm just doing that to show you how this works. The calculations are pretty straightforward, and we can do it rapidly and automatically on all the images. So what do we find when we come back to the Lewis framework? This is the first week that we deployed IFCB in June of 2006. Approximately 800,000 images that were phytoplankton, and here's the envelope that we see in the Lewis framework. Some indication that there's an expanded range and perhaps a contracted range relative to what Lewis suggested. Of course, this is 1 week in one ecosystem. If I fast, oh, sorry, 1 second. This is the like a histogram, 2D histogram, so you can see the relative frequency of different shape or morphological trait combinations in that one sample. Now, if we fast forward to the present, so this is the frequency distribution for all of the phytoplankton images from 2006 to 2021. And the conclusion I find from this is that Lewis's framework for the maximum surface area to volume that's typically observed seems sound, but when we look at this much greater range of phytoplankton in natural communities, we find that they're also occupying that sort of triangular wedge that he's labeled stout, simple shapes. So the range of surface area to volume is constrained, but not as narrowly as Lewis thought. All right, I just made a huge leap. I went from the 1 week to 16 years. Of course, there's a whole bunch of fascinating information in between. This is just for that first year that I showed you just a minute ago. This histogram here for the first week in June. We have multiple weeks in that year. At this point, the instrument was actually a prototype, so it was coming in and out of the water as we were advancing the engineering development, so we have gaps, but we can now produce these Lewis-style frameworks for, this is each year going by, week by week by week, until eventually it'll get to 2021. We could talk for a long time about all the information that's captured here. It's fascinating. There's all kinds of cool patterns, and it's kind of overwhelming and complicated to interpret. And as I was looking at these, I was reminded of the second paper that I want to turn to. Also important work, I think, put forward by Bill Lee in the late 90s. Bill proposed that based on individual measurements of phytoplankton from flow cytometry, which was sort of all the rage at that time, that we could calculate aspects of the diversity of the phytoplankton community based on these, I'll say, traits measured by conventional flow cytometry, so no imaging here. Bill was using measurements of individual cell chlorophyll fluorescence and light scattering. He put these into big wide bins and then computed and considered them as categories in this histogram sense. And then he computed what he called cytometric diversity, Shannon Weimer style diversity, and evenness. In this really lovely example, he showed 4 years of weekly sampling, flow cytometry sampling in Bedford Basin. And he was able to show that the cytometric diversity shows distinctive seasonal patterns, and also that the overall diversity was strongly driven by changes in evenness within this framework. I should mention that this analysis was focused on small phytoplankton, so pico to small nanoplankton. And one of the conclusions that he made from this study is both based on this time series and some spatial sampling that this kind of cytometric diversity in the cell properties is largely driven by changes in evenness rather than richness. So I was really intrigued by this framework. It looks like a good way of summarizing a lot of complex data to look at summary metrics of morphological diversity. And I wanted to explore whether this conclusion about the importance of evenness was extended to larger phytoplankton with these more complex morphologies. So back to the Lewis framework, you can do these analyses that Bill Lee did in multidimensional trait space. But I'm just going to simplify and look at the Lewis two-dimensional framework because I think it captures a lot of the important variability. So we can use the same kind of cytometric approach, only using these morphological characters from the images. And for this year, where we have histogram, 2D histograms for each week, I can then compute a diversity metric with weekly resolution for that particular year. We see patterns that are actually reminiscent of the cytometric diversity, high values in the winter, declining in the spring, relatively low, and then increasing through the summer. If you're like me, you're looking at what the heck happened in May? Like, did she collect bad data or what? I just want to show you that these metrics are really showing us something, summarizing a lot of variation in the environment. This is what the images look like from the second week in May, the third week in May, and then a couple of weeks later, beginning of June, all those long chain forming diatoms are gone. There's a few dinoflagellates and a whole bunch of nanoplankton. And that's reflected in the sort of traitscape there and in the diversity metric. Okay, so that was 1 year. We can compute these metrics for the entire time series, and the seasonal pattern emerges consistently higher in the winter, declining in the spring. Although you see that there's a ton of sort of event scale interannual variation where there are these spikes like that event that I showed you in May. And similarly, we can look at evenness based on these trait categories. And notably, the overall diversity and the evenness follow very similar patterns. So back to this question, Lee proposed the conclusion that evenness was driving the major changes in cytometric diversity. And we're finding with these data that a similar trend is emerging for larger phytoplankton based on their image morphology from images. I do want to say that the richness does vary, and it does seem to vary systematically. This is the diversity and evenness I showed you before. Richness based on category count, which is tricky, right? You have to be careful about this sample size and other challenges. I also, just to check this, I looked at different richness metrics like the Chow bias-corrected Chow-1 metric, and they both show similar patterns, seasonality, and changes in richness that are associated with those bloom events. But the impact on overall diversity is damped compared to changes in evenness. Okay, so I want to come to the third paper, this recent work by Fontana et al. In 2016, where they emphasize the important point that we're kind of oversimplifying with this cytometric diversity extending the cytometric diversity concept I just talked about, because these traits are not really categorical, right? That's an abstraction I was making by binning them. And we can get a much more in-depth view of morphological diversity if we incorporate the full distribution of trait values. It matters whether two traits are close in value versus far apart, even though they might be, have the same frequency distribution in two locations. So this isn't a new idea, but Fontana et al. Did some work to recommend a series of diversity metrics that incorporate the full distribution in n-dimensional trait space. The overall dispersion, which is just the mean distance of individuals from the centroid of all the measured in the sample. They proposed a new evenness metric that's based on the divergence between two distributions, where those distributions are based on the sum of all the pairwise distances among individuals in the sample, compared to individuals in the same size reference distribution, where they're evenly distributed in trait space. And then this kind of funny richness metric they call trait onion peeling, which is based on the sum of the surface areas of convex hulls, successive convex hulls, as you successively remove outer layers of individuals in the ND trait space. They propose that these individual level trait metrics should be more powerful, and I wanted to see if the conclusion that they could be more powerful predictors of variation, say in the aggregate level of community variability, was held when we look at these real data. So we can compute similar to the distributions I showed you, the full time series and annual cycle, like I showed you for the categorical metrics. These are just the full trait distribution dispersion and evenness. And if you recall, they look somewhat familiar to what I showed you earlier, high in the winter, declining into the summer, and the annual interannual variability and bloom events are quite evident in the variance. So, similar to first order to what we get with the categorical approach. So how would we assess whether these are better predictors or tell us more about the community? One preliminary example, I asked the question how well we could explain variance in the total community, the total aggregate biomass of the plankton community. This is a temperate zone where there are big changes in biomass seasonally, where it's highest in the winter. And how much of that can be explained by changes in diversity? An interesting question from an ecological point of view. When we look at the trait category approach, a small but significant amount of the variance in total biomass can be explained by changes in overall diversity. When we extend to that full trait distribution metric, so dispersion metric, a larger fraction of the variance in the total community biomass is explained. You see that summarized in the first two bars of this histogram. It's the same, the correlation coefficients, where the red bar is the dispersion metric that's based on the full trait distribution. Similar pattern emerges when we look at the evenness metric. A much higher proportion of the variance is explained when we consider the full trait distribution instead of this categorical approach. But then it looks like things break down when we look at the richness metrics. But it turns out this trait onion peeling metric is super sensitive to sample size and the edges of the trait distribution. That's a bit by design, but it leads to, I think, some real challenges with real data. I haven't emphasized that all the analysis I've shown you so far have included only weeks when I had more than 50,000 individuals observed. So the low sample size isn't influencing most of the metrics, which actually converge very quickly to unbiased estimates from the observations. For the onion peeling metric, I knew that the sample size, from Fontana's work, the sample size was very important. So I was randomly down selecting to the minimum number, 50,000, to make the computation of that metric. And with 50,000 observations, even if you do lots of resampling, there's a damped dynamic range and a bias low with that metric. If I make the criteria more stringent and only include the weeks when I have more than 400,000 individuals measured, it doesn't drop off too fast. We have a lot of data here. So there's still 544 weeks. And the histograms don't change appreciably, when I eliminate those 100 lowest sample size weeks, until I incorporate all 400,000 available, even sample observations into the trait onion peeling metric. And at that point, the dynamic range is perhaps more representative, and that metric actually explains more variance than any other in terms of the total biomass. But this is, maybe it's intriguing, and I'd be interested in if others have worked with this kind of metric and have ideas about how we might overcome this extreme sensitivity to sample size to make this actually a more practical metric that we can use to compare samples in a quantitative way. All right, so that was kind of a blitz, 50 years in 20 minutes. I just remind you that the results we have today expand the envelope for realized traits, more shaped traits in phytoplankton relative to what Lewis suggested in 1976. The evidence I showed you today suggests that the approach that Bill Lee put forward is powerful for looking at morphological diversity, and that some of the conclusions that Bill Lee found with cytometric diversity seem to extend to what we find in morphology of larger phytoplankton. And then finally, these full trait distribution metrics from individual, accounting for the actual values of individual traits in a large population, seem very promising and worth more study and perhaps incorporation into models. I'm interested in what folks think. And I will just leave you with an invitation to visit the IFCB dashboard if you want to see any of the billion images we've collected at MVCO, as well as those we're now collecting in spatial surveys that extend further offshore from the coastal observatory. Thank you.
Thank you, Heidi. That was this is like a game changer. A billion images to really give us some sense of traits. Questions?
Oh, sorry.
That was a great talk. It really shows the power of having huge amounts of data too. I was just curious on the evenness relationship. Does that correspond with blooms? In other words, if you get the unevenness associated with blooms, or is it more complicated than that?
I don't know if I mean I have to look more systematically to. There's a lot here, and there's a lot of, I'm sure I haven't explored yet. But I think to first order, my answer would be yes, there are changes that are associated with the blooms. I think if we go back to...
Yeah, I didn't really know.
It goes by fast, yeah. I know, but all of this stuff that looks like noise in this part, in this... sorry, I'll do it up here. It looks like, I was emphasizing this sort of systematic seasonality you see in the median through here, but all this variance here, that's real stuff associated with the blooms. In fact, whoops. What happened? You see the lowest trait evenness that happened twice? It looks like crazy here in 2009 and again in 2018. Anybody want to hazard a guess? What happened?
Hurricanes?
Phaeocystis. Do any of you know phaeocystis? So colonial phytoplankton with a gelatinous matrix, small cells that are embedded in this gelatinous matrix. They make these big colonies, and they apparently totally disrupt the rest of the phytoplankton community. That's been shown in other ecological studies, that it shows up really strongly in these metrics. So I think that that's one case study to show that that's going on.
Thank you.
Adam has a question.
So when people do these diversity analysis with sequence data, you often have this problem that evenness and richness are correlated simply because of the way the sample is taken. That you sequence a fixed number of sequences, and so if you have a high diversity, right, or high evenness, right, then you have a high diversity and vice versa. And so I wonder if it's the same thing with the images. Is there sort of did you take a sort of fixed number of images per time point or something like that, or is there a decorrelation in that sense between richness and? So
you're talking about like a rarefaction thing, right? If you don't sample deeply enough, is that what you're talking about? It was a little bit hard for me to hear you.
Sorry, it's mask and everything.
Yeah,
but,
I get.
Well, no. So the point is that you typically take 10,000 sequences, right? And so what does richness mean, right? Richness will be higher if the community is more even, right? And if it's less even, then richness will be lower. So there's sort of a mathematical built-in link between richness and evenness. And so I was just wondering about whether you have the same issue with imaging data, or
there's nothing.
Whether you feel like you exhaustively sample everything.
Yeah, so we're of course we're not exhaustively sampling anything, right? We'd have to measure every organism in the ocean to in the limit, and there's nothing conceptually different about this from other observations of individual properties that you would use to measure. There's no magic here, so the same issues apply. We have a lot of observations, so most of the metrics except that onion peeling one are converging to, they're robust estimates. We've really sampled very deeply for the standard diversity metrics. But richness, of course, in this case, the way that it's an abstraction of the resolution of the categories and other sort of a conceptual exercise. As long as you do it consistently within a data set, I think it's comparable. But it's definitely, as I mentioned, it's trickier to interpret. And those three metrics, the are interrelated, as you say, mathematically, right? You compute the evenness by the ratio of the 0th order Hill number to the exponential of the 0th order in the denominator and the 1st order in the numerator. So you're right. Sorry, that was long-winded. And I'm love to hear other people's thoughts about how to disentangle some of this and make sense of it.
I have a much more simpler question. So for those of us who assume the spherical cow or the spherical phytoplankton, from your time series data, can you tell us, and considering we use this spherical phytoplankton to calculate carbon biomass, can you tell us when our measurements or our calculations might be most strongly biased, like seasonally, from your time series data?
Oh, well, in this location, the answer to that is unequivocally in the winter time. But that's because this system has these blooms of diatoms that they're all elongated, right? It's very, and it has to do with this trade space that Lewis was talking about. If they're, yeah. So I don't know. I can go back to the 2011 results. I think you can see some examples. You see those like bright yellow lines going up at different times in the time series. Like there's some here where there's like a whole bunch of stuff going up like this, and you see it sometimes in the winter. That's where you would have, if you have a cylindrical shape that's just all different lengths, right? So it's a particular species or genus of phytoplankton that is blooming at that time. And if you're going to, convert that to a sphere, you're going to get a completely different relationship between the maximum length and the surface area to volume. So if you care about, how susceptible they are to a grazer, maybe the maximum length is what's important. If you're trying to, if you're concerned about nutrient uptake, it may be this. None of those are any different, right? They all have the same, basically the same surface area to volume. As they go up that, they're just getting longer and longer. So there's some other selection pressure for them to get longer. That's not helping them increase their surface area to volume.
Yes, thank you. Do you have an opinion on the need for a community standardized image processing measuring library so that there could be standardized measurements across various instruments so they could have comparative data sets?
Well, maybe that's a bit of a loaded question. Jessica might have something to say about it, but I am absolutely an advocate for extreme care in these assessment of these size assessments. You can get biased and very different results depending on the details of the algorithm and in the calibration of pixel to size. And we put a lot of careful and work into getting these algorithms as unbiased as possible for the IFCB images. I don't think you can just extend that without validation to a different type of image or different images from a different kind of instrument. So I think that careful work needs to be done. And then, kind of cross validation where you have overlapping size ranges among different instruments will be important too.
Well, thank you, Heidi. I think we could probably do a whole session about how we could use some of these traits in incorporating them, the ecological function of them.